All right, guys, I'm going to attempt to talk you through how DNA becomes, I should say becomes, but works with RNA to become proteins, which we know are what make up our genes. And then from there, you get all the unique characteristics that make up, well, that make you, you, okay? So where I'm going to start first is with the DNA. So all I'm going to do is kind of draw this for you, and then hopefully talk it through. So obviously you have in the nucleus, we pretend this is the nucleus, you have your DNA, okay? And your DNA is in that beautiful double helix shape, right? And then you have certain bases, what are called nitrogen bases, that are naturally paired together. Okay, so I'm going to draw just a couple examples of that. And based on the order of these things, that's what's going to determine the proteins that are made in the near future here. Okay, so we'll say you have DNA, and if you remember, with DNA, you have the 4 different types of bases. You have your adenine, you have your thymine, guanine, and last but not least, cytosine. Now, what makes them special, like I said, is the sequence or the order that they're paired in. Now, in terms of, okay, you have your DNA, so what happens next? You have to remember that DNA is really just the cookbook, okay? So it's a cookbook, or it's meant to be the instructions. It doesn't mean it physically does anything. Instead, it's the blueprint for the RNA to really make everything happen, to make this construction of a protein, construction of a gene, to make that happen. That's where the DNA comes in, okay? So this first phase, what happens is within our nucleus, and I'll actually write that just so you know the where part. So if this is our nucleus, what happens is you get this little enzyme. And I know in our notes last time, I attempted to draw kind of like a Pac-Man type thing, okay? So here we have our Pac-Man. The whole point of him is that the Pac-Man, his job is to go in and say, all right, well, this DNA is what DNA is what we're going to try to interpret. So what the Pac-Man or the enzyme needs to do is to take this double helix shape and then basically unzip it into single helix. Okay, so all I mean, and I'm going to jump to my next slide here, is that Pac-Man looking dude is going to come along. There he is. And he's going to break this DNA strand like that. Okay, so instead of being just the single strand, now he's going to start breaking it apart into something like what you would see here. Okay, so he's literally taking the double helix that was all nice and twisted, and he's going to start breaking that apart. From there, what's happening, and keeping in mind we're still in the nucleus, from there what you're going to have is our messenger RNA. Okay, so I'm going to say messenger, or mRNA. Put that into parentheses, messenger. Now, what makes the messenger RNA important, and what its role is in all of this, is the messenger RNA and then is going to come along, and it's literally going to make, so pretend that this is our messenger RNA. I'm just going to put a big M. What he's going to do is it's going to say, okay, I'm going to read this helix. So I'm going to read this single helix here and single helix here. Now from there, if you remember, because we already broke apart the double helix, the messenger RNA says, okay, now what can I group with each of these? So for example, if the messenger RNA comes along, he's going to say, all right, if I want to group something with this helix, what would it be? And when I say group something, what I mean are those nitrogen bases. So it's going to say, all right, well, if the first one is that red, so what I'm looking at is this guy. So I'll erase these other arrows. So I'm just looking at this first little base. The messenger RNA's job is to say, all right, well, what would pair with that? And so the messenger RNA is going to begin to transcribe or read the DNA. So this actual phase in this whole process is what's called transcription. And all that means, like if you transcribe, you write something down, and that's all that's really happening. So the mRNA, what it's doing is it's reading and then more or less recording what it's seeing. So the messenger RNA here is going to say, all right, well, we have 2 adenine, just pretend we have 2 adenines, then a cytosine. So then the messenger RNA says, okay, well, we got to start grouping stuff with that. And so it'll come up with, as you know, our base pairs. You have your guanine always with your cytosine. Now the difference here is that if you remember, initially we had this. We had our adenine grouping up with our thymine, right? Well, what happens is the messenger RNA, so this blue strand here, what that's doing, it's a copy, right? So if something's a copy, it's not the original. So what's going to happen with messenger RNA is there's one big difference between what the messenger RNA is creating from reading and transcribing the DNA. So it looks at this blueprint and says, okay, I can make that, but there's one change I have to make, and that would be this T. Okay, so I'm going to actually get rid of that T and then add a U. So the U stands for uracil. Now, uracil is another nitrogen base, okay? So uracil is what's going to be grouping with, sorry, that's a crappy arrow, but it's going to be grouping with adenine, okay? So uracil is taking the place of thymine in the messenger RNA. So as the messenger RNA starts going, this little Pac-Man looking dude starts going all the way down the strand of DNA, the messenger RNA is going to continue to interpret or transcribe or write down or figure out what are all the matching pairs for the DNA. And once it gets done, so now I'm going to draw, we're going to pretend this is our messenger RNA, right? And then I'm going to go like this. So keeping in mind that this is a single strand now, because you had the Pac-Man guy ripping apart the double helix. And so right now what happens is you're left with adenine. These are your bases. Cytosine, guanine. And last but not least, remember, because this is a copy, it's not the original. The DNA is the original. You now have uracil in place of thymine. Okay, so instead of A grouping with T, you now have adenine grouping with uracil. All right, so your messenger RNA, which is right here, messenger RNA is now going to head outside the nucleus. Okay, so we're going to pretend that way down here in the corner, this red dot I'm attempting to color. We're going to pretend that that is the nucleus. So now the messenger RNA is gone. It's saying, all right, now I have this strand, this copy. Now I got to figure out how is this copy going to begin to build what are called amino acids. Okay. And it can only do that with the help of 2 things. First, you have your ribosome. We're going to pretend this is the ribosome. It's a very original color right there. Now, the cool thing about ribosomes is you got them all over the place. So meaning outside of the nucleus. And so the messenger RNA is then greeted by a ribosome. Now, within the ribosome is more like the location, but within it, it has what are called rRNA. And very fittingly, the R in rRNA stands for ribosomal. Okay, so this messenger RNA is on its way to the first ribosome it sees. Okay, and it does that because the ribosome's job is going to be to break this into its parts. So now it's going to figure out or interpret, or a better word for interpret would be this. I'm going to jump ahead here if I can. There you go. A better word for that would be translate, which is why this phase is fittingly called translation. So basically you have that messenger RNA strand, which is now visiting our ribosome. And from there, the ribosome is saying, okay, I'm going to start breaking this into codons. So it's going to start looking at this in terms of, all right, for every 3 of these little random bases that I can find, what does that really mean? Or what is that telling me? And so by our ribosome and ribosomal RNA, taking a look at our messenger RNA, what's going to happen is it's going to say, all right, I can see right here that I have maybe like an adenine, a uracil, and a cytosine. Like that's the first 3. And then what happens is the rRNA says, okay, if you have that sequence of code or that sequence of bases, well, then what that means is that a specific amino acid is going to be built or created. But in order for that to happen, you need some help. And fittingly, you have help in the form of something called transfer RNA. Now, transfer is the technical, but sometimes I've heard people call it just transport. Either way, I'm fine with it. And the reason being, I mean, transfer is what the T stands for, but the importance of these tRNA is that they are transporting amino acids. So what happens is you literally have these transport or transfer RNA. And we'll say that the first one says, all right, if you have on this, if the sequence is, all right, there's going to be an adenine, and then we'll pretend a cytosine, and then I don't know, a uracil, we're just pretending, then that would mean that you have a specific type of amino acid. So the tRNA says, hey, wait a minute, that's the sequence. I can match up with that. So then the tRNA is going to head over here, and it's going to literally bring an amino acid. So let me write that here, amino acid. It's going to say, hey, you need this based on the sequence that you're translating. That means this. So that would be like, the rRNA is kind of like, it's kind of doing the work in terms of it's saying, all right, well, this is like a different language to us. I have to translate what this means. And then once it figures out what something means, it then messages the transfer RNA and says, hey, this particular sequence is asking for this amino acid. Can you go out and find it? So the tRNA are constantly on the move, bringing different amino acids. Okay, so if you remember, for every, so we got, here, let's go backwards a little bit. So we've got our DNA, then we got our mRNA, and then that's going to end up working with our RNA, our RNA in the ribosomes. Then from there, we have our tRNA, which is going to help us create amino acids. Now, why that's important is because as those amino acids are being grouped, the amino acids are going to create none other than the building block of everything, proteins. Now, proteins are important because they are what dictate the genes we have, characteristics we have. In terms of, we've been talking about the COVID-19 vaccine. The antibodies your body builds are literally, well, first off, the vaccine itself is a form of mRNA, as we talked about, but the antibodies that are built are actually a type of protein. Like antibodies are protein, it's just that they're a specific type. So if we go back really quickly through all of this, remember you have your DNA, which is alive and well in the nucleus. You've got your enzyme that comes along and says, all right, we need to start building here. You got this DNA, what does it mean? So the enzyme starts breaking it down, okay? It starts unzipping our DNA, which then, as soon as that starts happening, the messenger RNA says, all right, I gotta go to work. And so it begins transcribing or figuring out what does the DNA tell it. As it's doing that, it's making or building individual copies of both strands or each single helix. Okay. However, as you know, it is a copy. mRNA is a copy of something. So you have evidence of that due to uracil. So uracil is the only, like, if you see uracil right away, your brain should be like, okay, that is no longer DNA, that is RNA, because uracil is replacing thymine. From there, your messenger RNA says, all right, let's go to work. We have this message. We know what we got to build. Now I need some help. So I need some more RNA to begin translating this message that I just copied from the DNA. So out it goes to the ribosome, and then through the process called translation, these adorable little transfer RNA begin helping the messenger RNA and our RNA. They help it to begin building amino acids. As those amino acids start getting grouped together, they start building proteins. And then as those proteins start building together, you get genes, you get, I'm just going to say characteristics that make you, you. So hopefully this makes sense. Our quiz on Tuesday, what I'm going to do is I'm going to break it into some possible questions, and we will review, so don't panic. Would be something like, you know, okay, where is DNA found? That could be a question. What are the 4 nitrogen bases within DNA? That could be a question. Once the enzyme begins unzipping the DNA, which type of RNA begins to play a role? That answer would be messenger RNA. Well, maybe a short answer question might be, all right, if you look at a single strand, how can you tell if it's DNA versus RNA? Well, if you see this, a U instead of a T, that's a giveaway that that is no longer DNA. That is the copy that was made by the RNA. Another question might be, uracil replaces which base? That would be thymine. Another question might be, once the messenger RNA leaves the nucleus, where does it go? That would be the ribosome. Question might be, the ribosome is the location, but what actually does the work to translate the messenger RNA? And that would be the rRNA. And last but not least, the tRNA is busy bringing over what? It searches the cell to find what? And that would be amino acids, which eventually will build proteins. So if you have questions between now and Tuesday, obviously ask. Just Slack me. Otherwise, I'll be in touch. So good luck, guys. Hope you're all well, and I'll see you soon.